basically, if we have a population and we know how likely it is that a dominant allele is being inherited and a recessive allele is being inherited, we can plug it into this equation and predict what the frequencies should be in the next generation of offspring. If no change is happening in this population and they are not being affected by any evolutionary forces, then these allele frequencies would remain the same generation after generation after generation. So this is one way to kind of test that. So let's say we have pea plants and we know that 60% of the alleles are dominant and 40% are recessive. So they should add up to 100% because an allele can only be dominant or recessive. So that's what the P plus Q equals 1 part is referring to. So P stands for the dominant allele and Q stands for the frequency of the recessive allele. And the combination should add up to 1 or 100% basically. So if you just had 1, if we were just told 60% are dominant, then we would be able to figure out what the frequency of the recessive alleles are, just knowing that I have to add up to 100%. So we are given both, however, and this equation is set up so that P squared represents the frequency of homozygous dominant individuals, 2PQ is heterozygous, and Q squared homozygous recessive. So we would need to put this in decimal format, but basically we have the dominant allele 60%, so 0.6, and the recessive allele would be 0.4 since it is 40%. And we just plug it into this equation. So 0.6 squared is 0.36, 2PQ, we got 0.48, and Q squared is 0.16. These aspects of this equation just represent these genotypes here, and we can convert that into percentages. So that's basically 36%, 48%, and 16%. So in the next generation, 36% should be homozygous dominant, 48% should be heterozygous, and 16% should be homozygous recessive individuals. So that is what we would get if the population was not changing in any way. So if we actually observed the gene frequencies in the next population, we could compare it to the prediction. So let's say we actually went out and just looked at the genes for everyone in the population, and we found 30% homozygous dominant, 60% heterozygous, 10% homozygous recessive. That's what we actually found. That definitely differs from our prediction. So what was it, 30, 60, and 10? So our prediction was 36%, 48%, and 16%, but the actual numbers were different than what the prediction would be. So that means that this population is changing somehow, and that evolution is occurring in this population because the gene frequencies are changing from what they should be if everything would be consistent here. So keep in mind that everything adds up to 100%. So if one gene changes, then it will change the others. But basically, if you see any difference at all, then the population would be evolving. It would have to be exactly the same as this prediction for it to be in Hardy-Weinberg equilibrium. If it is not in equilibrium, then it is evolving. It's basically kind of one or the other. If we're looking at a population here, it will either be in equilibrium or it will be evolving. It is difficult to actually see Hardy-Weinberg equilibrium in a population. There are rare cases where a population could be in equilibrium, or maybe a certain gene is in equilibrium and the gene, that specific gene, is just not really changing in how it's being inherited. Most populations probably will be evolving or going through different kinds of change in some way, whether people are moving out of the population, new people are coming into the population. So for the most part, we would see populations not in equilibrium. But there are a few cases where that could happen, like with this study that was done with blood types in the UK. They have a slightly different blood typing system where they use N and M alleles, and people can be homozygous dominant, heterozygous, or homozygous recessive for these blood type alleles. So they use Hardy-Weinberg equilibrium and basically use that equation to predict what percentage of people in the population should have these blood types based on this. And blood typing was an interesting thing to study because there is a large population. At that time when they were doing this, there wasn't a huge amount of immigration. We didn't see enough heritable mutations in blood types to really significantly affect which blood types are being inherited. There isn't a particular survival advantage for having a certain blood type over another. And mating is pretty random when we're talking about blood types specifically. People aren't generally picking mates for their blood types. Maybe some people might, but for the most part, people are not purposely selecting mates on blood type. So they went out and actually documented the what we could observe in the population, or what we actually observed there. And they observed data that was really close to the Hardy-Weinberg equilibrium equation. So their observations really very closely fit in with this model. So that was showing that maybe blood type alleles were not really changing in frequency in this population at this time period. And this population was in equilibrium when we're talking about blood types here. If you had a Hardy-Weinberg problem and say you are just given that the dominant allele is 70% of the population. So what would Q be? Q would be 0.3. It would have to be 30% in that case. And if we filled it in to P squared, 2PQ, and Q squared, it would give us these frequencies. We would have 49%, 42%, and 9% for homozygous dominant, heterozygous, and homozygous recessive. And if we actually went out and observed how frequent these genotypes were, and we got 49%, 42%, and 9%, would this be in equilibrium or evolving? It would be in equilibrium because it's basically the same as our prediction. But if we got like 60%, 20%, 20%, what's going on here? This population would be evolving because the actual observed genotypes differ from what we would see if it was in equilibrium.